Hi, Bio 101. It's Dr. Lennon again with part 2 of mitosis in eukaryotic cells. We left off here at the cell cycle, and we'll start back up here as well. We talked about the idea that interphase is actually about 90% of the life cycle of the cell, which means that the mitotic phase and cytokinesis is really only 10% of the life cycle of a cell. Interphase itself is broken into 3 distinct phases. The first one is G1, or the gap phase 1. In this phase, the cell is just functioning normally. It's a normal cell. It's functioning, it's growing, it's making proteins, etc. G1 is actually the longest phase, and it's followed by the S phase, or the synthesis phase. That's where DNA is synthesized or replicated. So at the end of S phase, you actually have your DNA replicated. Each chromosome is made up of 2 sister chromatids, and you have twice as much DNA in the cell. After S phase, we move into G2 phase. In G2, the cell is still functioning normally, but it's also getting ready for mitosis. So the chromosomes have been replicated. We have sister chromatids making up each chromosome. And in G2, we're getting ready for the M phase, or the mitotic phase. You'll notice in this online lecture, if you download the PowerPoint, you'll see that there are links to go to other resources, and I encourage you to do that. In truth, I don't dare click on that link right now because I'm not quite sure how it will behave, and I want to get this lecture recorded for you. So properly, this is part 2. Mitosis is conventionally divided into 5 phases. The phases of mitosis are prophase, prometaphase, metaphase, anaphase, and telophase, or telophase. So those are the phases of mitosis for our purposes in Bio 101. After mitosis, we go into cytokinesis. And cytokinesis actually overlaps with telophase or telophase. Remember, cytokinesis is where we split the cytoplasm of the cell. If you go to Mastering Biology, you'll find a nice video of mitosis. Now, it's really important to remember that these phases of mitosis are human kind of inventions to some degree. Mitosis itself is a fluid process. So what we do when we look at the fluid process of mitosis, just like everything else that humans do, is we try to break it down into distinct little chunks. Chunking, as you know from learning, makes things easier. Breaking things down into smaller pieces makes them easier to understand. So you should know as we talk about these phases that they are a little bit fluid. A trick that I have used ever since 7th grade for remembering the order of the phases of the cell cycle is IPMAT. So my sister was studying to be a biology teacher. She's 7 years older than I am, so I was pretty little when she was doing this. And she taught me to remember the phases of the cell cycle with IPMAT. I for interphase, so that's the part that's not mitosis. P for prophase, P for prometaphase, M for metaphase, A for anaphase, and T for telophase. If you find that sort of thing helpful, go ahead and use it. I'm sure my sister would be excited to hear that it's helping you. So we're going to start looking at the cell cycle. This is right before we go into mitosis. This is G2 of interphase. If you look at the pictures on the left, you'll see a real cell on the top. You'll see the nucleus in the middle, and we see part of the cytoskeleton showing here in yellow. On the bottom, we see an artist representation of this phase. Something to remember as we look at these phases is these pictures are going to be what happens by the end of the phase. So by the end of G2 of interphase, this is what we see. Our chromatin has been duplicated. We have a nucleolus. We have an intact nuclear envelope. And our centrosomes have been duplicated as well. So we would see two centrosomes. We're going to see what those do in a moment. A centrosome is really a microtubule organizing center. So the centrosome is involved in organizing our microtubules, and we're going to see how those are important also in a few minutes. So there's our nuclear envelope is intact. Now the chromosomes have been duplicated or replicated, but we don't really see them as individual chromosomes. If you look at the real cell, you don't really see them as individual chromosomes. They're not condensed into those really characteristic chromosomes that we're used to seeing in cell division. So here's the first phase of mitosis, and it's prophase. Again, the normal cell at the top. You can start to see some little snake-like things in there. So we start to see our chromosomes becoming visible. If you can't see it, you might want to dim the lights and look closely. In the bottom, we can see the artist's representation of it. Our nucleolus disappears, which makes sense because as we learned about in Chapter 4, the nucleolus is a very active part of the nucleus. And as we go into mitosis, the nucleus becomes sort of inactive, is a good way to think about it. There's not a lot of activity with the DNA because the DNA is packed tightly into chromosomes. So our nucleolus disappears. The chromosomes start to condense and become visible, and you can see that here and here. The sister chromatids are joined at the centromeres and start to become visible. I would say you'd have to look very, very closely. Our centrosomes, which you can see actually pretty well on both of these, but the artist representation is a little easier. So here's a centrosome, and here's a centrosome, not to be confused with a centromere. The centrosomes start to move towards the poles of the cell. So you can sort of see them here in this picture as well. So they're starting to separate, and they're going to separate and have one on either end. The mitotic spindle, which is made up of microtubules, begins to form. So if we see here in the artist representation, the spindle is a meshwork of microtubules between our centrosomes, and it starts to form. And you can sort of see that here as well. The yellowish green color are our microtubules. Our mitotic spindle is made up of microtubules. It's required to divide the chromosomes, and we're going to see how that works. It's produced by the centrosomes, and those again are the microtubule organizing centers. They organize the microtubules, and you can say that the microtubules kind of grow out. That's an origin point of our microtubules. And they contain a pair of something called centrioles in animal cells, which are involved in organizing those microtubules. Here's prometaphase, which you may or may not have heard about in high school. You really start to see those chromosomes, and if you look closely, you can start to see that they're made up of chromatids. Our nuclear envelope breaks up and disappears. We really don't see a distinct nuclear envelope here. Our chromosomes become even more visible and condensed. Each sister chromatid has something called a kinetochore. So at the centromere, where the little waist is, we see a protein on the outside surface of each sister chromatid. So the outside surface of this sister, the outside surface of that sister. It's a specialized protein structure called the kinetochore, and that's where our spindle microtubules are going to stick to the sisters. During prometaphase, the microtubules attach to that kinetochore and start to jerk the sister chromatids around. So we start to see those moving. So here's metaphase. I kind of think of between prometaphase and metaphase, it's almost like getting a group of rowdy children in line. We kind of know that takes some time. If you've ever tried to get little kids to stand in a straight line and stand still and stay there, it's difficult. So in prometaphase to metaphase, that's really what we're doing. We're getting the sisters in line. It tends to be one of the longest stages of mitosis. Our centrosomes at this point, by the end of metaphase, are at opposite poles, and they have this nice spindle between them. So they're connected by a fully formed mitotic spindle. Chromosomes are still made up of sister chromatids, and they're lined up at what we think of as an imaginary plate. So the metaphase plate isn't an actual physical thing. It's just the center of the cell, kind of like the equator of the cell. By the end of metaphase, the sisters are lined up there. And they're lined up there due to the mitotic spindle attaching and moving them around until they're lined up. So the microtubules of the spindle attach to those kinetochores and move them around. Here's anaphase. Anaphase is usually pretty quick. And anaphase, we're popping the sisters apart. Once you pop the sister chromatids apart, they become sister chromosomes or daughter chromosomes. Either one works. So it's pretty quick usually. The sister chromatids are pulled apart from each other. They become daughter chromosomes. So here are the now daughters, from sisters to daughters. It's a little odd. So now we've got two individual chromosomes. There's one chromosome, there's the other chromosome, and they should be identical barring any weird mutations. In anaphase, we're going to start moving them towards the poles of the cell. So the daughter chromosomes are pulled to opposite poles by spindle microtubules attached to their kinetochore proteins. By the end of anaphase, both ends of the cell have equal and complete sets of chromosomes. So we've got one copy over here that's going to be pulled to this end, and another copy over here that's going to be pulled to this end. Something else that's happening in anaphase is the cell is elongating. It's getting longer in preparation for cytokinesis. So our cell's getting longer, we're pulling the sisters apart, and we move on to telophase. So in telophase or telophase, we're actually getting the nuclei. The two daughter nuclei form with the nuclear envelopes, and we start to get nucleoli. Which means in telophase, even before we had two fully separated cells, we had active DNA. So the nucleoli form, which means we're having activity in the nucleus. DNA is being used. Genes are being used to make RNA and ribosomal RNA, etc. The chromosomes decondense, which means, again, if they decondense, they're now available for use, and they become less distinct. At the end of telophase, mitosis is complete. Cell division is not complete yet, but mitosis is. So our nuclei, we have two nuclei now, we have split our nucleus, which means we've finished mitosis. To complete cell division, we have to complete cytokinesis. So it's going to begin during telophase, and it forms a cleavage furrow in animal cells and a cell plate in walled cells. The cleavage furrow is shown right here and right here. It's almost like we took a belt and just pulled very, very, very tightly, and we're squeezing those cells with a cleavage furrow. We're going to squeeze that belt and pull until our cells separate. A little different in plant cells, and we'll take a look at that as well. So here are the phases of the cell cycle, and you're going to be looking for these in lab this week. You're going to be looking at plant cells. They're a little easier to see. I would at least print off the last couple pages of this lecture, and you have the PowerPoint version as well, and bring those in as a reference to use when you're looking at these cells under a microscope. It is expected you're going to be doing this individually, so you're going to be looking at cells and trying to figure out what they are. You may also bring your book if you would like. So here are the phases. Those will hopefully be helpful. Here's what they really look like. And again, you at least want to print this part out. Here are our phases in plants. and the same phases in animals. The same thing is going on. We just have the fluorescence microscopy here, and we have our standard compound light microscopy with the plants. We could have done either way. So it just shows you what they would look like. Alright, so make sure you print this part out from the PowerPoint. It's also available on Moodle. Bring this to lab. Watch this as many times as you would like, and I'll see you in Bio 101.